Welcome, deep divers. Have you ever paused to think about the incredible journey your morning toast or a glass of yogurt takes once it enters your body?
It's quite something, isn't it?
Yeah, we're talking about carbohydrates. They're the essential fuel for most of the world. Their metabolism is this, well, finely tuned symphony, and frankly, it's nothing short of incredible. Our bodies process them in such complex, elegant ways, and today we're pulling back the curtain on that intricate process. Okay, let's unpack this.
Let's do it.
This deep dive is your shortcut to truly understanding this vital biochemical process from a clinical perspective.
Absolutely. And whether you're, gearing up for an exam, trying to make sense of how what you eat impacts your body, or you just have an insatiable curiosity about your internal machinery, this deep dive is for you. Think of this as your audio guide through, well, a core chapter of biochemistry. We're going to follow carbohydrates right from the moment they hit your plate.
Okay.
Through the intricate steps of digestion, absorption, and all the way to how they actually fuel your cells.
And it gets even better. We're not just tracing pathways. We'll be exploring what happens when things go wrong. I think, common discomforts like lactose intolerance.
Yeah.
Or how conditions like diabetes are managed at this really fundamental biochemical level. We'll even dive into a truly life-threatening biochemical emergency like cholera.
Yeah, that's a critical one.
And discover how understanding carbohydrate transport literally saves lives. So get ready for some compelling, real-world, clinical examples that will make these concepts truly stick.
Oh.
The carbohydrate landscape. What are we eating? So let's start at the beginning. What exactly are we putting into our bodies when we talk about dietary carbohydrates? What are the main players here?
Well, the major carbohydrates in our diets are primarily starch, lactose, and sucrose.
Okay.
And these aren't just common. They collectively represent the largest source of dietary calories for most people globally. But from a biochemical standpoint, they're quite distinct, and those differences are really key to how our bodies handle them. Well, starch, for instance, is a complex polysaccharide. That means it's a huge chain of sugar units. Think of it like a massive branched tree made of glucose molecules.
Wow.
We see it as amylose and amylopectin, where these glucose units are linked by specific bonds, the I-1,4 and the I-amylore-4-phels glycosidic bonds. You find these in grains, tubers like potatoes, various vegetables.
Got it.
Then there's lactose. Much simpler, a disaccharide, meaning just two sugar units linked together. It's glucose and galactose, connected by a Mi-1,4 glycosidic bond, and is found pretty much exclusively in milk and dairy products.
Right, the milk sugar.
Exactly. And finally, sucrose, our common table sugar, is also a disaccharide, but this one's glucose and fructose linked by an I-1,2 glycosidic bond. This one's everywhere, fruits, honey, and it's a very common additive in countless processed foods.
So the key insight here is how these subtle differences in their molecular handshakes, those specific bond types, they dictate entirely different digestive requirements, and as we'll see, can lead to very different clinical outcomes.
Yeah.
But no matter how complex or simple they start, the overarching goal for all these diverse forms, once they hit our digestive system, is to convert them into their simplest building blocks.
Precisely. We need to break them all down into their constituent monosaccharides, that's glucose, fructose, and galactose, because that's the only way they can actually be absorbed from our gut into our bloodstream.
Like breaking down Legos.
Exactly. Think of it like needing to break down a Lego castle into individual Lego bricks before you can use them to build something else.
Okay. And while we're talking about dietary carbs, we can't forget about dietary fiber. What's its role, or maybe lack thereof, in all this?
That's a crucial distinction. Dietary fiber refers to polysaccharides that our human digestive enzymes simply cannot break down. They just can't touch them.
So they pass through.
They pass through our small intestine largely untouched. Their fate is quite different, impacting our colon in ways we'll touch on later. But for now, the key is they don't get absorbed for energy like other carbs.
That idea of indigestible components really makes me think of something I once heard about starch blockers. Was there really a period where people thought you could just block starch digestion for weight loss?
Oh, absolutely. It's a fascinating historical footnote in biochemistry, really. Many years ago, there was this concept of starch blockers marketed for weight loss.
Right.
The idea was that certain proteins, often from beans, could inhibit amylase, the starch-digesting enzyme. So the thinking was, you could eat all the starch you wanted and it would just pass through undigested.
Fewer calories absorbed.
Exactly, effectively reducing calorie intake. Unfortunately, the early versions didn't really work effectively in the human body.
Why not?
Well, the stomach's low pH often inactivated the inhibitor protein, and our bodies are incredibly efficient, producing amylase in such abundance that it was just hard to overcome.
So, a good idea in theory, maybe, but the biochemistry proved more complex in practice.
Exactly. But here's where it gets really interesting. Current research is actually revisiting their potential. Newer preparations are showing promise, carefully designed to overcome those earlier challenges. It's a classic example of how understanding the fundamental biochemistry, even when it leads to initial failure, can eventually inform effective, real-world health interventions. 2. The digestive journey. Enzymes at work.
Okay, now that we've mapped out the major players on your plate, the real biochemical adventure begins as they journey through your digestive system.
Yeah.
Let's follow these carbohydrates on their intricate path.
Indeed. And digestion of starch actually kicks off right there in your mouth.
In the mouth already.
Yep, with an enzyme called salivary α-amylase. This enzyme is what we call an endoglycosidase. That means it attacks internal α-amylase bonds within those massive starch molecules,
Okay.
breaking them down into smaller pieces called α-dextrins. So it's like taking a long rope and chopping it into shorter segments.
Makes sense.
But this initial work is short-lived. Salivary amylase is quickly inactivated by the highly acidic environment of your stomach. Gone.
So, a powerful start in the mouth, but quickly shut down by stomach acid. It sounds like our bodies must have a robust backup plan in the small intestine then.
Precisely. The real heavy lifting for starch continues in the small intestine. Your exocrine pancreas steps up. It secretes bicarbonate to neutralize that stomach acid.
Crucial step.
Very. And along with that, a vital enzyme called pancreatic amylase into the duodenum, the first part of the small intestine. This enzyme picks up where salivary amylase left off, continuing to break down starch and glycogen.
What does it produce?
It produces disaccharides like maltose, trisaccharides like maltotriose, and other small oligosaccharides called limit-dextrins. These are unique because they contain those IR16 branches from the original complex starch structure that amylase itself can't actually break.
It's fascinating how robust this system is. You're saying our bodies are so good at this that even if our pancreas isn't quite up to par, starch digestion might still be okay.
That's generally right, yeah. Amylase activity in the gut is typically so abundant that it's rarely the limiting step in digestion. Even in cases of significant pancreatic dysfunction, like with severe pancreatitis or cystic fibrosis, starch digestion often isn't as severely affected as, say, protein or fat digestion. There's just so much amylase around that it can usually compensate to a large degree.
Interesting.
And we even have pharmacological tools that interact directly with this process. Take acarbose, for example.
Right, the diabetes drug.
Exactly. It's used in type 2 diabetes, and it works by blocking both pancreatic amylase and certain enzymes at the brush border. This effectively slows down the rate at which carbohydrates are broken down and absorbed into the bloodstream.
Helping with blood sugar spikes after meals.
Precisely. It helps manage those post-meal blood sugar spikes. However, the trade-off is that more undigested sugars reach the colon, which can lead to side effects like, well, flatulence and diarrhea. It's a balancing act for patients.
So the initial attack by amylase breaks the big chains down. But the final granular work happens further along, almost at the doorstep of absorption.
That's it.
The journey continues to the intestinal brush border for the final breakdown into individual sugar units.
That's exactly right. The small intestine is lined with these tiny, finger-like projections called microvilli, which form what we call the brush border. It creates a huge surface area.
Okay.
And attached to these microvilli are specialized enzymes called brush border disaccharidases. Think of these enzymes as highly specialized keys, each designed to unlock a very specific sugar molecule, making them ready for absorption.
A whole toolkit.
A whole toolkit, yeah. We have several key players here. There's glucoamylase, which is an exoglycosidase. That means it chops off individual glucose units from the end of a chain. It's specific for MSLR4 bonds and can digest things like limit-dextrins down to isomaltase. Then there's the sucrose isomaltase complex. This is a fascinating enzyme with two distinct active sites. One site hydrolyzes sucrose table sugar and most of the maltose. The other site is crucial because it's the primary enzyme for breaking down those tricky α-1-6 bonds in isomaltose.
The branches amylase couldn't handle.
Exactly.
Yeah.
Tackling those branched structures amylase couldn't handle, plus it has some additional maltase activity. Together, these two handle about 80% of the small intestine's maltase activity. They're workhorses.
Wow.
Another interesting one is trehalase. This specifically hydrolyzes trehalose, which is a sugar found in things like insects, mushrooms, and fungi. It has an unusual all-1 glycosidic bond.
Never heard of it.
Yeah, it's less common in Western diets maybe, but a deficiency in trehalase can cause severe gastrointestinal distress. I actually recall a case where a patient with trehalase deficiency became incredibly sick after eating wild mushrooms.
Oh, no.
And was initially suspected of poisoning. It just highlights how specific these enzyme deficiencies can be and how important they are for specific dietary components. And finally, a particularly important one for many, many people, the Beck-glycosidase complex, also known as lactase-glucosylceramidase. Its lactase catalytic site is absolutely crucial. It hydrolyzes the 1-4 bond in lactose.
Breaking down milk sugar.
Exactly, breaking it into its constituent glucose and galactose. The other site, interestingly, digests glycolipids, but it's the lactase activity that most people are familiar with, or perhaps, unfortunately, familiar with.
So a whole team of specialists lined up to break down these sugars right at the point of absorption. And are these enzymes found everywhere in the small intestine, or do they have specific zones?
That's a great question. They actually have varying activity levels along the length of the small intestine. Sucrose, isomaltase, and lactase activities are generally highest in the jejunum, that's the middle section.
Where most absorption happens.
Yeah, where a lot of digestion and absorption happens.
Yeah.
Glucoamylase activity, however, actually increases as you move further down towards the ileum, the last section.
Why is that?
It provides a sort of last-ditch effort, a final chance for any remaining starch oligomers that might have slipped past the earlier digestive steps. It's like having a final quality control check before the finish line. 3. When digestion goes awry. Clinical insights.
Okay, this brings us perfectly to the real-world impact of these intricate processes. Let's step into the waiting room, so to speak, and look at some scenarios that illustrate what happens when carbohydrate digestion or absorption goes wrong.
Good idea. Our first scenario involves Denise V. She's a 20-year-old exchange student experiencing, gastrointestinal bloating, abdominal cramps, and diarrhea, especially after consuming dairy products.
Sounds very familiar.
Yes, these are the classic symptoms of lactose intolerance. For Denise, who didn't consume much dairy back in Nigeria and then adopted a dairy-rich diet here, it's characteristic of adult hypolactasia.
Meaning her lactase levels dropped.
Exactly. This is actually the normal physiological decrease in lactase activity that most of the world's population experiences after childhood, as the biological need for milk digestion naturally wanes.
So it's not really a disease for most people.
In that sense, no. It's
Sure.
important to distinguish it from congenital lactase deficiency, which is a rare, severe inherited condition where infants can't digest lactose right from birth. Or secondary lactase deficiency, which happens due to intestinal injury from things like gastroenteritis or malnutrition that temporarily damages the cells that produce lactase.
Right.
The mechanism of lactose intolerance is quite straightforward. Without enough lactase enzyme, undigested lactose travels all the way to the colon.
Okay.
There, bacteria go to town on it. They ferment it, producing lactic acid and gases, hydrogen, carbon dioxide, methane, which leads to the bloating and cramps.
Oh, the gas.
And that undigested lactose and lactic acid also exert an osmotic effect. They pull water into the bowel lumen,
Causing the diarrhea.
causing the characteristic diarrhea. Diagnosis is often just made by eliminating dairy and seeing if symptoms improve, but a hydrogen breath test can confirm it by detecting that bacterial gas production.
And management.
Usually involves reducing or avoiding lactose or using commercially available lactase supplements, just like Denise V found helpful. It's also crucial to ensure adequate calcium intake from other sources if dairy is significantly reduced.
Makes sense. What's next?
Then there's Nina M., a 13-month-old baby experiencing severe distress screaming, lots of gas, and a distended abdomen, especially after consuming fruit juice.
Fruit juice. Not dairy this time.
No. This strongly suggests a problem with either sucrose digestion or, perhaps more commonly, fructose malabsorption. Fruit juice is often very high in sucrose, which contains fructose, or sometimes just high in free fructose itself.
Right.
If that fructose isn't absorbed properly in the small intestine, same story gets to the colon, where bacteria metabolize it, producing gas and causing those painful symptoms.
So it's not just dairy that can cause problems, but even seemingly healthy things like fruit juice if there's an absorption issue. Is this fructose issue common?
It's actually more common than you might think. A lot of people don't realize that more than 50% of adults might have some difficulty absorbing high doses of fructose, say around 50 grams, and even over 10% can't completely absorb 25 grams.
Wow.
So it's why some people feel digestive discomfort after large amounts of fruit juice or certain high fructose foods.
You mentioned dietary fiber earlier, explaining it's indigestible by our own enzymes. So what does this all mean for someone managing their blood sugar, like Deborah S with her diabetes? This seems like a really critical connection.
It absolutely is, especially for Deborah. While our enzymes can't digest fiber, colonic bacteria can. They metabolize soluble dietary fibers like pectins and gums found in oats, beans, or apples into gases and short-chain fatty acids. Things like acetic, propionic, and butyric acid.
Do those do anything?
Yeah, these fatty acids can actually provide a small amount of energy for the cells lining the colon. This bacterial action is also why increasing fiber intake can sometimes cause some initial bloating and flatulence as your gut microbiome adjusts. But for Deborah, the benefits of fiber are profound. Certain soluble fibers, like pectins and the glucan you get from oats, have been linked to lowering blood cholesterol by binding bile acids. But more importantly for diabetes management,
Yes.
they can slow the rate of absorption of simple sugars. They effectively create a speed bump in the gut.
Slowing things down.
Exactly. This prevents those rapid, dramatic spikes in blood glucose levels after meals, which is absolutely crucial for managing diabetes. This is where the concept of the glycemic index comes in handy.
Right, the GI index.
It's an indicator of how rapidly blood glucose levels rise after eating a specific food. Understanding high versus low glycemic index foods, along with the fiber and fat content, is a cornerstone for managing conditions like diabetes.
Can you give us a quick example, high versus low?
Certainly. Foods like, say, cornflakes and potatoes generally have a high glycemic index because their carbohydrates are very rapidly digested and absorbed. Quick spike. In contrast, things like yogurt and skim milk, even with their sugar content, often have a particularly low glycemic index, maybe due to their other components and slower overall digestion.
Interesting.
So, incorporating soluble fiber, think oatmeal, beans, apples, and choosing lower glycemic index foods, maybe pasta over potatoes sometimes, is excellent advice for Deborah S. to manage her blood glucose more effectively and avoid those sharp peaks.
Very practical.
Finally, let's look at a real biochemical emergency: cholera.
Okay.
This is a severe diarrheal disorder caused by the bacterium Vibrio cholerae. It secretes a potent exotoxin.
And what does the toxin do?
This exotoxin dramatically increases levels of a signaling molecule called CAN-MP inside intestinal cells. This, in turn, does two bad things. It diminishes the normal absorption of sodium, other anions, and water.
So less fluid getting in.
Right. And at the same time, it fiercely stimulates the secretion of chloride and water into the gut lumen.
Oh, wow. So you're losing fluid from both ends, essentially.
Oh, exactly. The result is massive, life-threatening fluid loss, sometimes exceeding a liter per hour. This leads to rapid dehydration and shock.
That sounds absolutely terrifying. How does biochemistry offer a lifeline in such a severe crisis?
This is truly an aha moment in medical history. The genius of oral rehydration therapy, or ORT,
RDT,
with glucose and sodium, is its simplicity and stunning effectiveness. The crucial insight was realizing that the cholera toxin does not affect the NAIR+-dependent glucose transporters in the intestinal cells. They still work.
So there's still a way in.
There's still a way in. By co-administering glucose and sodium in a carefully balanced solution, these unaffected transporters can still pull glucose and sodium, and, crucially, water follows them into the body even while the toxin is raging.
Incredible.
It partially corrects the severe fluid and electrolyte deficits. Adding amino acids can enhance this effect even further. ORT has saved literally millions of lives globally. It's a powerful testament to understanding these fundamental transport mechanisms. 4. From gut to body. Absorption and transport of monosaccharides.
Okay, so we've broken down the carbs. Now, how do these essential sugars, glucose, galactose, and fructose, actually get from our intestine into our bloodstream and ultimately to the cells that need them? It's a journey from the gut lumen to, well, literally every cell in your body.
Indeed. Once digested down into their monosaccharide forms, these sugars must be transported across those absorptive epithelial cells lining the small intestine. From there, they enter the blood for distribution to all the tissues that need them for energy or maybe for storage.
How does that happen?
There are two primary transport mechanisms involved in this absorption process. First, we have the Na plus I-dependent glucose transporters, or SGLTs.
SGLTs. Yeah.
These are like active gates located on the luminal side of the intestinal cells, the side facing the gut contents. They actively concentrate glucose and galactose from the gut lumen into the cell, even if the concentration inside the cell is already higher.
How do they do that? Against the gradient.
They achieve this by essentially coupling the transport of the sugar with the movement of sodium ions, which are moving down their own steep concentration gradient into the cell.
So it's like a tiny biochemical leverage system using the sodium flow.
Exactly. Think of it this way. Imagine a revolving door, the SGLT. There's a huge push of sodium ions wanting to get inside, and the door only turns if a glucose molecule comes along for the ride.
Got it.
So the energy of sodium rushing in helps literally pull glucose into the cell. And this low intracellular sodium concentration, which drives the whole process, is maintained by a pump, the NO plus AK plus ATPase on the blood side of the cell. This pump actively uses energy, ATP, to pump sodium out. That makes the SGLT process a form of secondary active transport.
Secondary active transport.
Okay. And you see similar SGLTs in your kidney cells too. They're vital for reabsorbing glucose there, so we don't just pee out all our valuable glucose.
Right. What's the other mechanism?
The second type are the facilitative glucose transporters, or GLUTs. This is a whole family of proteins, GLUT1 up to GLUT5 and beyond. They don't bind sodium. Instead, they act more like passive channels, allowing sugars to move down their concentration gradient from high concentration to low.
So no energy needed directly.
Correct. On the serosal or blood side of the intestinal cells, GLUTs move glucose and galactose from the now high concentration inside the cell out into the lower concentration in the bloodstream.
And fructose, does it use these too?
Fructose, interestingly, primarily uses a specific transporter called GLUT5 for facilitated diffusion to both enter and leave the absorptive epithelial cells. What's intriguing is that fructose seems to be absorbed more rapidly when it's ingested as part of sucrose compared to when it's consumed as a standalone monosaccharide.
Why would that be?
It's likely due to how the sucrose is digested right there at the brush border by sucrase, presenting the fructose and glucose right next to their respective transporters, GLUT5 and SGLT1, GLUT2. Sort of an efficient handoff.
Okay, so different sugars use different gateways. But once they're in the bloodstream, do all cells take up glucose in the same way? Or does it vary?
That's a fantastic question, and the answer is definitely no. It varies a lot. The GLUT family of transporters isn't just in the gut. Different tissues express different GLUT protein isoforms, and this reflects their unique metabolic roles and needs.
Like what?
Well, for most general cells, glucose transport usually isn't the limiting factor for glucose metabolism. These cells either have a high concentration of GLUTs or they have GLUTs with a very high affinity for glucose, a low coulombum ensuring a steady supply pretty much all the time.
Okay.
Red blood cells, for instance, which rely heavily on glucose, have a high concentration of GLUT1. This ensures they get adequate glucose even when blood glucose levels might be a bit lower. They're always hungry.
Makes sense. What about the liver?
The liver is different. It primarily has GLUT2, which has a relatively high coulombum. Remember, a high coulombum means it has a lower affinity for glucose.
So it's not grabbing glucose unless there's a lot around.
Exactly. The liver primarily takes up and converts glucose into other energy storage molecules, like glycogen or fat, only when blood glucose levels are high, like right after you've eaten a meal. This is absolutely crucial for its role in maintaining overall blood glucose homeostasis. It ensures the liver doesn't hoard glucose when other tissues might need it more urgently, but it efficiently processes any excess after a meal.
Very clever system.
And here's a vital connection back to conditions like Deborah S's diabetes. In muscle and adipose tissue, that's fat tissue glucose transport, is significantly stimulated by insulin.
Ah, the insulin connection.
Yes, insulin acts as a critical signal. It triggers the movement, the recruitment of GLUT4 transporters from storage vesicles inside the cell up to the plasma membrane, the cell surface.
So insulin opens the gates, basically.
It puts more gates on the surface.
Yeah.
This dramatically increases glucose availability for energy use and glycogen synthesis in muscle, and for making fatty acids and glycerol in adipose tissue. So without enough insulin, or if the cells become resistant to insulin signal.
Like in type 2 diabetes.
Exactly. Those GLUT4 transporters remain largely stuck inside the cell. Glucose can't get in efficiently, so it accumulates in the blood, leading to hyperglycemia, a classic hallmark of diabetes.
And what about the most glucose-dependent organ of all, our brain? How does that crucial fuel get past the blood-brain barrier.
That's probably the most critical transport of all. The brain's capillaries have extremely tight junctions between their cells, forming what we call the blood-brain barrier, the very selective, very protective gateway.
Right.
Glucose has to pass first via GLUT1 transporters in those endothelial cells that form the barrier itself, and then it needs to pass through GLUT3 transporters to actually get into the neurons.
So a two-step process just to get into the brain cells.
Essentially, yes. And what's critically important here is that at low blood glucose levels, let's say below about 5 millimolar or around 80 milligrams per deciliter, the rate of glucose transport into the brain actually becomes the rate-limiting step for brain function.
Meaning the brain doesn't get enough fuel.
Precisely. This is why you see those neuroglycopenic symptoms. Lightheadedness, dizziness, confusion, slurred speech. Potentially even coma when blood glucose drops too low, hypoglycemia. The brain simply isn't getting enough fuel fast enough, and it's a stark, immediate reminder of its absolute dependence on a steady, adequate glucose supply. Outro.
Wow, what an incredible and intricate journey we've taken today. Really, from the major carbohydrates on your plate, starch, lactose, sucrose, through their meticulous breakdown by all those specialized enzymes.
Salivary amylase, pancreatic amylase, the brush-bordered disaccharidases.
Right. In your mouth, pancreas, and intestinal lining. All the way to their final absorption as single sugars into your bloodstream and onward to fuel literally every cell. We've seen how these fundamental processes influence everything from dietary choices for someone managing diabetes like Deborah S.
Yeah, think about fiber and glycemic index.
To common discomforts like lactose intolerance for Denise V, and even life-saving interventions using ORT in a biochemical emergency like cholera.
It truly puts so much into perspective, doesn't it? Understanding these fundamental biochemical pathways really helps us appreciate the nuances of diet, how various health conditions manifest themselves, and importantly, the rationale behind therapeutic interventions.
Definitely.
And this raises an important question for you listening. How might your awareness of carbohydrate digestion and absorption, having gone through this, maybe change the way you approach your next meal or snack? Or even how you understand symptoms you or perhaps others around you might experience?
It's powerful stuff, literally fueling life itself. Thank you so much for joining us on this deep dive into the absolutely fascinating world of carbohydrate metabolism.
It was a pleasure.
And a warm thank you from the Deep Dive team for tuning in.